So I was very cheered up by your initial introduction actually earlier, when you said that part of this aim of this meeting was to get people in from different areas, because I was feeling like a bit of a fraud before that, because I'm not an expert in CFS, but I do have some expertise in doing medical imaging, and that's what I'm hoping to talk to you about today. And hopefully by getting across some of the methods we use, and particularly some of the kind of more conceptual approaches that we take to studying diseases and drugs.
Sorry, Matt, can you just move your microphone up a little?
Oh, sorry. Come on. Is that better?
Yeah.
Yeah, good. We can hopefully, by thinking about some of the concepts and the approaches that we use, will spark some ideas and maybe even some collaborations in this group. So, as was said, we're a consortium of universities and the MRC. So we were originally set up by GSK. The lab site was set up in 2007. GSK spent a lot of money on it, and then 4 years later decided they didn't want it anymore, as these companies do. The lab was taken over by King's, UCL, Imperial and the MRC as a consortium, and they formed this new kind of fairly unique entity called Imanova, which is a private company but owned by public bodies. And that's how we've been operating since 2011, and we do a mixture of academic and commercial research. So all that was true up until literally a week ago. So a week ago we actually signed a deal with an American company called In Vitro, who work in a similar kind of area. They tend to specialise more in the very early phase clinical trials, doing animal work, preclinical work. So we're now wholly owned by In Vitro. We're not owned by universities anymore. Universities decided that they wanted to get rid of the risk of owning us. It's a period of change for us. We're not sure how it's going to pan out in the future, but that's how it is from now on. So up until, for the last 6 years, we've been operating this kind of model where we do some commercial work, mostly for the pharma industry. And the idea is that commercial work hopefully subsidises some of the academic work, and the more kind of basic science academic work allows us to develop methods that we can use and translate into the commercial work and increase our commercial offering. We mostly focused on neuroscience. We do some other stuff, oncology, a bit of respiratory, a bit of immunology stuff. So we're kind of semi-evil drug company, but also semi-nice. We're Robin Hood, basically. We steal from the rich drug companies and give to the poor academics. That's how we like to think about it. So we have some very nice facilities. We've got 2 cyclotrons for producing radioactive isotopes, a lot of chemistry labs, 2 PET CT scanners, 2 MRI scanners, and 1 new combined PET and MRI scanner, various assorted radiation and blood sampling equipment, some nice blood labs, good IT and analysis infrastructure, and some basic clinical areas as well. And the way we like to think about this is more our commercial work when we're looking at tends to be looking at new drugs, testing out new drugs, is this. So you have a new drug, you put the drug into the patient, and you can see the drug in the blood system. Hopefully the drug gets to the tissue that you want it to get to, in this case the brain, or this kind of general scheme applies to any other organ as well. The drug hopefully binds to a specific target in the brain or any other organ, maybe sets off some kind of second messenger system. There's then perhaps a broader tissue response or network response in the brain, which leads to some physiological or behavioural response, and that hopefully leads to a cheesy stock photo of a happy patient, which is what you want, a good clinical outcome. So traditionally, if you're doing this kind of drug development research, your options in humans anyway for looking at this kind of thing were really restricted to the beginning and the end of the chain, if you like. You could do pharmacokinetic studies and look at the kinetics of the drug in the blood, and you could do kind of more PD, pharmacodynamic studies, and look at the effect on physiology or behaviour. What this lab was set up to do originally by GSK is really to get rid of that grey area in the middle and really be able to see the whole chain. And you can think about disease pathways in a kind of similar way. Now, obviously, this is a very generic and simplified way of thinking about diseases, but I hope you'll find it useful. I hope to convince you that it's useful. So, you have your original etiology, whatever that might be, environmental, genetic, other, viral, which leads to some kind of primary pathology. That primary pathology might produce biomarkers you can see in the blood or in other body tissues. Secondary pathologies as a result of the primary pathology, broader, say, network dysfunctions in the brain or broader dysfunctions in other organs or body tissues, physiological behavioural symptoms, and that eventually leads to the full clinical presentation. So again, the idea of what we do when we try and work in this kind of area of characterisation of disease states is to figure out that middle section which is otherwise obscured. And in these cases, we try and think about it in terms of imaging biomarkers or high biomarkers. And one of the aims of everything we do is really to identify robust and reliable imaging metrics that can serve as biomarkers in various diseases. And the way we do this is with two different technologies, really. So positron emission tomography is very good at looking at the kind of more molecular early stages of that pathway, and MRI is generally better suited to looking at the later stages of the pathway. So this is how to do PET in one slide, in 15 seconds, so it's pretty brief. But you have to generate some radioactive isotopes in big particle accelerators called cyclotrons. Normally, we use carbon-11, fluorine-18. You can use others. There are several different other isotopes that people have used. Carbon-11 and fluorine tend to be the most common. You have to do some chemistry, so you have to take that radioactive carbon atom, remove a carbon atom from another molecule that you're interested in, and put that isotope on it and radioactively label the molecule, which is tricky because you have to do some chemistry with these things, and everything is radioactive. You have to quality and safety check it, because the next thing you're going to do is put that radioactively labelled substance into a patient. You put the patient into a PET scanner, and the PET scanner gives you 3D images of the distribution of radioactivity in the body. Now, the kind of information you get from the PET scanner depends crucially on the molecule that you've chosen to use. So, for instance, we've got a mouse pointer here. So, a dopaminergic molecule, something that binds to dopamine receptors, will give you an image like this of the very dopaminergic areas in the brain, because the molecule will bind to receptors and accumulate in areas where there are a lot of dopamine receptors. So there are lots of different PET radiotracers that have been developed. You have things like raclopride and PHNO, which are selective for dopamine receptors. You have carfentanil, which is an opiate, and obviously selective for opiate receptors, mu opioid receptors particularly. You have things like flutometamol, which binds to beta amyloid, which is a protein which accumulates in the brains of Alzheimer's patients. And you have something called PBR111, which binds to translocated protein as a marker of microglia and macrophage activity, which is thought to be a general marker of neuroinflammation. The different kinds of PET studies, so a simple version is a biodistribution study where you just look where does the radiation go in the body. And this is important if you're developing a new drug and you need to know, well, does it actually get into the brain or the tissue of interest. The brain is particularly tricky because of the blood-brain barrier, which screens a lot of stuff out. So if you have a drug which you imagine think is neuroactive, and you want to confirm that it does actually get into the brain, PET is the way to do it. The more sophisticated kind of studies tend to be called receptor occupancy studies, and this is where you might do several PET scans on different patients under different treatment conditions, and you try and derive really nice, solid quantitative measures of the relationship between the dose of a drug and the proportion of receptors that you occupy in a particular, say, brain region. So if you have a drug where you think from your animal studies your therapeutic window is going to be above 70% occupancy of receptors, you can do a dose ranging study, find the dose in humans that will give you that level of occupancy and therefore be effective. As an example, I've got some a study that we did a couple of years ago using PBR111 PET imaging, which is, as I said, is a general marker of neuroinflammation. So this was in MS, and in MS patients you tend to find elevated levels of PBR111 binding across the board, particularly elevated in areas in the white matter in the brain where there's lesions. This is healthy controls here. So in MS you tend to get these characteristic lesions. This is the brain of an MS patient. The white areas are the lesions in the white matter, and you see that PBR111 binds very nicely to the areas around the lesions. It makes sense that you get more neuroinflammation around these lesion processes. What was also nice as well is that we found that the PBR111 binding was also quite tightly related to a general measure of debilitation in MS. So the more symptoms somebody has, the more neuroinflammation we can detect with PBR111. The other technology that we use is MRI, and this is my specialist field. So MRI is a lot easier in a lot of ways than PET. It's generally a lot less invasive, it's a lot safer, there's no nasty mucking around with radioactivity, it's cheap, or it's cheaper than PET anyway, and it's a very flexible modality, so you can look at 3D structure of the interior of the body, you can get quantitative markers of disease progression, you can look at brain activity and brain connectivity, and these last two are really my specialist field, functional MRI. So this is a little schematic of a functional MRI experiment. It's actually my visual cortex sliced open at the back there. So functional MRI exploits differences in the magnetic properties of oxygenated and deoxygenated hemoglobin. When a brain area is active, it requires more oxygen. The vascular system dumps oxygen into the area, produces a local change in the magnetic properties of that area, and that's what we can detect with functional MRI. So you can see the brain working in real time. This is a little study which was actually really smoking hot off the presses. It was published yesterday, that we did looking at e-cigarettes. It's just an illustration of a functional MRI study. So we realised a couple of years ago that nobody had ever had people smoking in an MRI scanner before. And this is important to know that, because cigarette addiction is not just about the nicotine. Nicotine administered through other forms is not a desperately addictive drug. Nobody gets addicted to nicotine patches or nicotine gum if they weren't originally a smoker. But there's something about the whole sensory and behavioural aspect of smoking, the delivery system of the nicotine, the smell, the taste, the habit, which is very, very powerful in driving the addiction, as well as the nicotine, or in combination with the nicotine. So it's important to understand both sides of the coin. And there's been a lot of functional brain imaging studies on nicotine, where they've injected it or delivered it orally or via patches or things like that. But nobody's ever allowed, I imagine it's probably because hospitals really don't want you smoking cigarettes in their £2 million medical scanners. There's all kinds of safety issues, obviously, but we realised a little while ago, well, why don't we try it with e-cigarettes, these little electronic devices? And there's issues with that as well, because they tend to have metallic components inside, and MRI scanners are big magnets, and those things don't mix very well, but it's possible. So we did it, and we found the main result actually was big activity in the left motor cortex, and that tells you that people tend to move their right hands a lot when they smoke, which is not very surprising, but still, you know, part of it. More interesting were these results in the kind of thalamus and striatum, these very dopamine-rich, reward-related, addiction-related regions in the centre, showing that the action of smoking and the sensation, the sensory aspects of smoking, were kind of intrinsically rewarding. So that's two examples of separate PET and MRI studies that we've done. The kind of studies that we actually like to do are what we call multimodal imaging. And, PET and MRI become much more powerful when you put them together. So you can do these studies separately. You can put people in a PET scanner and then give them a short break and put them in the MRI scanner, or even do it on a different day. But we've just taken delivery of a brand new combined PET MR system. Well, it was delivered in about May, but we're still figuring out how it works. So we've done a few scans on it. It takes a while. We've done a few scans on it, but we're still kind of commissioning it, basically, and validating it. And when you put these two modalities together, you really start to get some good insights, I think. So this is another study, again in MS, really a kind of follow-on from the previous study. Again, looking at neuroinflammation with PBR111, but this time particularly looking at the symptom of depression that you tend to get in MS. And so MS patients tend to be depressed at a higher rate than you would see maybe in other chronic illnesses. It's thought that there's something particular about MS which causes the depressive symptoms as well. And we were interested in, particularly in an area of the brain called the hippocampus, in the medial temporal lobes, highlighted in blue there. And in the hippocampus, we get higher measures of neuroinflammation with this PET ligand in patients than controls. And neuroinflammation doesn't tend to differ too much in other nearby areas like the thalamus. And we also see that the hippocampal neuroinflammation measure is very tightly correlated with Beck depression inventory scores, one of the main clinical measures of depression. So that's the PET side. Then we did some FMRI scans on these people as well, and particularly looking at the connectivity of the hippocampus. So what these images show is the relationship between the hippocampus and the rest of the brain. Now every brain area has a kind of network of other areas that it preferentially communicates with. And in the healthy controls here, you can see the hippocampus in very bright yellow-white there, and you can see that it communicates with the striatum, ventral striatum, the bits of the putamen and ventral caudate there. This area in the medial frontal lobe is very significant. Various bits in the temporal lobes, and this area in the posterior cingulate cortex as well, also the anterior cingulate cortex. And you get more or less the same pattern in MS patients. I guess the most obvious decrease is in maybe the posterior cingulate cortex there, but that pattern's more or less preserved. When you start combining the two sets of data, so the PET imaging data and the fMRI, looking at relationships between the two, is where it gets interesting though. So this is a little hard to get your head around, but this is showing the relationship between hippocampal neuroinflammation and the connectivity of the hippocampus. So there's a negative relationship between hippocampal inflammation in some of these areas, in the blue colour scale there, and a positive relationship with others. What's really interesting is that when you do the same thing with their depression scores, you get a very, very similar looking network. And this is the conjunction between the two there. And what this allows us to do is perhaps generate, one more thing. Yes, the area with the best relationship between all these three different variables turns out to be this area in the subgenual cingulate cortex, which has a nice relationship with BDI score and also with the neuroinflammation in the hippocampus in its connectivity to the hippocampus. And it turns out the subgenual cingulate is really the key area in a lot of depression research, and it's particularly it's even the main region that people tend to target with deep brain stimulation when they're trying to treat very chronic, long-term, treatment-resistant depression. So this allows us perhaps to start thinking tentatively about a little model of what might be going on, similar to the kind of response pathways that I was talking about at the beginning. So there's hippocampal pathology related to the MS, which leads to neuroinflammation, which leads to these network effects that we've seen in the MRI scans, particularly in subgenual singular dysfunction, and this may be what's causing the symptoms of clinical depression that you see co-occurring with MS. And this is all very interesting, but we hope it's useful as well, because, okay, we can't cure, unfortunately, the underlying pathology of MS at the moment. There is no cure, but perhaps we can design additional interventions which can have an influence on other stages of the chain. If we can break that chain at any point, if this model is correct, then we'll hopefully alleviate some of the symptoms of depression. So maybe we might try some anti-inflammatory drugs, maybe we might try influencing this network in some way, or deep brain stimulation on the subgenual singular. So understanding diseases in this way gives us additional targets or additional approaches, perhaps, to designing interventions. So, didn't run over too much, good. What I've hoped to get across here is that the kind of approach that we use and the kind of work that we do using these two techniques. So PET and MRI are both powerful techniques. A lot of places use them completely separately, but we think combined is where they really deliver in terms of powerful insights into the organisation of these issues. So very, very useful in characterisation of disease states, better characterisation of disease, better chance of designing targeted interventions to disrupt these response pathways. And the neat thing is that if you have an idea for a potential intervention, you can then evaluate that intervention using the same methods. So you want to use some, say, anti-inflammatory drugs to treat MS. Well, okay, let's do a PET study with measures of neuroinflammation and test the drug as well. So it becomes a kind of almost an iterative process, yeah, refining and refining ideas. Lots of issues with doing this kind of research. Patient recruitment is difficult, that kind of thing. Probably the biggest issue is cost. MRI is relatively cheap. PET is incredibly expensive, but that's getting somewhat better with the new combined scanners. They might be a little bit cheaper because you only have to pay for one scan, but that's still a major barrier. So I hope that sparked some ideas and given you a different perspective on some of these things, and thank you very much.
Yeah, thank you, Mark.